So first, we've got to talk about our experiments for chapter 13. We're going to talk about chapter 13 and get into chapter 14 as much as we can. For chapter 13, the first person we talked about was Frederick Griffith. Okay? Thing was about like what he figured out. Transformation is taking place. Didn't know what, but he knew transformation takes place. So he figured out that transformation happens. And so how he did this, all right, is he used some bacteria and he used some mice, all right? And what he did is he took a living S strain, all right? And when he took this living S strain and gave it to a mouse, it was all bad, okay? Okay? And then he gave a living R strain. When he gave that living R to the mouse, he was happy. The mouse lived, everything was cool, like peachy keen. Then he gave a heat-killed, what the heck is heck? Heat-killed S, and do you remember what happened? He's cool. Okay? Then he gave heat-killed S and living R, so I'm just going to put LR. And what happened next? So when he gave the living S, it died. When he gave living R, it lived. When he gave heat-killed S, it lived. But when he combined these two things together, all of a sudden that mouse died again. Meaning, somehow when these two combined together, it turned back into this and killed it. Okay? You have a question? Oh, or are you just moving your strap? Oh, I thought you had a question. I was like, alright, cool. Alright, and so he just left off with that and said, this is really cool, guys. I found out something's being transformed. I don't know what it is, but I'll let someone else do that. Next. Okay, we're not going to go into the details about his experiment, but do you remember what Oswald Avery figured out? The thing being transformed is what? It's nucleic acids. Okay, so transformation nucleic acids. Okay? Then we get to, and so after this, everyone is like, that's awesome, like, I'm really proud of you, but it's kind of irrelevant because we already know that genetic material is proteins based on all the stuff we know. So, but you did, it's kind of a waste, but cool story, bro. So then came along Hershey and Chase. Okay? What they did is they said, all right, we don't think it's proteins, and we really want to investigate this. And so we're going to take these things, and we're going to make radioactive forms of this element so that we can label them and track them. We're going to put them in a virus, and with the way a virus replicates, because it's an obligate intracellular parasite, it's going to inject its genetic material into E. coli bacteria. I can then look at that E. coli and see, did it take up this genetic material? If it was in sulfur, it's going to be protein. If it was in the phosphorus, it's going to be nucleic acid. And so what they did is they looked at radioactive sulfur. Okay? And if it was radioactive sulfur, it's going to be found in proteins. Okay? If it was radioactive phosphorus, it's going to equal DNA. Okay? And what they found at the end, it was this one. Because remember, DNA is made up of these long repeating chains of nucleotides. Alright? And every single nucleotide has 3 parts. Okay? Every single nucleotide has this nitrogenous base. Every single nucleotide is going to have this sugar. And do you remember what else it has? A phosphate. Okay, that's going to be a PO4 minus or P for phosphorus. So that's where that phosphorus is at. So if you get confused on, oh, was sulfur with DNA or it was phosphorus with DNA. Just remember, DNA is made of nucleotides. A nucleotide has a phosphate, so it's phosphorus. Okay? And remember that backbone of DNA is sugar, phosphate, so phosphorus, phosphate. Cool story, bro. Okay? And then we get to the next guy. Okay? So, Hershey and Chase said, hey, guess what, guys? Nucleic acids are actually genetic information. And so that was kind of big, because that would kind of went against everything that everyone had believed up to that.
Are you recording this?
What?
Are you recording this?
No. So you pay attention. Okay?
Are we recording?
I don't care what you do. You're not my kids. Okay? So, Chargaff. What Chargaff figured out is he said the percent of A's is going to equal the percent of T's. And the percent of C's is going to equal the percent of G's. I don't know why, but that's what I found out. All the time, percent of A is going to equal the percent of T's, percent of C's is going to equal the percent of G's. All right? And so I've got that. And so for an example of a problem like that, if I have like A's, C's, T's, and G's, okay? And I say, let's say we looked at this whole sequence of DNA and we found 36% to be A. 36% of my DNA is A. What's the percent composition of all the other nucleotides? What would be my next step? T equals 36 because we know A is going to equal T. So T is going to equal 36%. Okay? And now what? Okay? So what's 36 plus 36?
72.
72. So I know of my DNA, 72% of the composition is that. Meaning C plus G equals 28%. And C equals G, so 28 divided by 2 is going to equal what those are. What's 28 divided by 2? C equals 14%. G equals 14%. Okay? That's how you figure that out. Okay? So Chargaff is the one who figured out the percent of A's equals the percent of T's, the percent of C's equals the percent of G's. Don't know why, but that's what it does. All right? And then we come along to our most recent ones. Okay? Okay, Rosalind Franklin and Maurice Wilkins. You remember what they did? Okay, they were the ones who kind of did the groundwork to figure out the structure of that DNA because they were doing X-ray diffraction.
Are they the ones that died?
She is, yeah. I don't know when he died, but she is, yes. She's the one who did the majority of the work and everything, and all of that. It's because of her. And then came along
And Franklin is that her?
Rosalind Franklin, yeah. Watson and Crick. Okay? They're the ones who kind of get the credit for the structure. Structure of DNA. They also are the ones who figured out why this happened, because they said A pairs with T and C pairs with G. That's why you have equal percentages, because you've got a double helix, and on one side I've got A, and on one side I've got T. Every time I have an A, it's going to pair with a T. Every time I have C, it's going to pair with a G. That's why there's equal percentages, because I have a double helix. All right? And they're complementary to each other. And they also figured out that G and A equal, and C and T equals pyrimidines. Okay? And that's the experiments. Next we go into replication. Okay? Any questions on this? You're Gucci? Yeah. Okay. Before I erase this, anyone need it? Sorry, I'm not. Alright. Finish yours, Rebane. you guys watch the inauguration second period?
Yeah.
Hang in there, guys. We got like 14 days till ski week. Something like that. 14 or 15. Like school days. Because there's like 3 more weeks, isn't there? Yeah. Sorry, I just gotta erase this, otherwise it wrecks my markers. Okay. So the next thing we're going to talk about is going to be DNA replication. Okay, DNA replication. And where's this going to happen? So this happens inside of the cell, all right? And we got inside of our nucleus and everything, and it's going to happen during the S phase of the cell cycle, because that's when DNA gets replicated, thus replication, all right? Or synthesis. So if I want to do this whole DNA replication thing, the very first thing is I've got to look for a little specific sequence, and something's got to open it. In DNA replication, what's the thing that unzips it? Helicase. Okay? Helicase is going to be the thing that unzips it. Okay? So helicase. Okay? It's going to break hydrogen bonds. And unzip DNA. Okay? So, if I made this beautiful DNA strand, let's say I've got this beautiful DNA strand. Okay? I would have an enzyme right here. This is helicase. Okay? Then, now that I've done this, I've got 2 things to kind of help protect my DNA while I'm doing this entire process. One of them is in front of the helicase. Okay? What's the thing that's in front of the helicase that's going to prevent overcoiling? Do you remember what it's called? Topoisomerase. Okay? So I have something in front called topoisomerase. Okay? So that's going to prevent overcoiling in front of helicase. Okay? So that's going to prevent overcoiling in front of helicase. So if I had that, it'd be like this little enzyme right here. Okay? That's in front of it, prevents this overcoiling. Then, I've got something else that's going to stop it from rebinding. Okay? Do you remember what those are called? That actually binds it. Okay? That binds the fragment. They get abbreviated often. It's called single-stranded binding proteins. Okay? Or SSBPs. Okay? So, okay, so single-stranded SSBPs, okay? They prevent the DNA from rebinding. Okay, they prevent DNA rebinding. So now if I looked at that over here, it'd be like these little things. Okay, I'd have them like right here, right here, right here, right here, right here, right here. These little single-stranded binding proteins prevent SSBP. This right here is topoisomerase. Okay, and so now I've done all of this. I need to lay something down so I can start doing this. So let's say right here, let's say this is a 3' end. Let's say this is a 5' end. So this would be 3'. This would be 5'. Okay. And now I've got to make these new strands. Okay. But before I can do anything, I've got to lay something down. What do I have to lay down? I've got to lay down a primer. And what does that? Primase. The enzyme primase does that. All right. So the first thing I've got to do is primase. Okay. It lays primer. So it's going to look for a specific sequence and it's going to lay down a primer. Primer is going to be an RNA sequence that says start here. Okay, it's going to be an RNA sequence that starts here. So I've got two strands here. I've got one that's one strand that's going to be a leading strand, and I've got one strand that's a lagging strand. So if I've got this strand right here, it's going to lay a primer right there, all right, of RNA. So this strand's going to go this way. This strand right here has to lay several primers. Okay? So I've got this thing. On one strand it only lays one primer. On this strand it's got to lay several primers. Okay? And primase does that. And then something's going to come along now, and it's going to start adding complementary nucleotides. Do you remember what that is? DNA polymerase 3. Okay? So now that I've laid these primers, DNA polymerase 3, it's going to lay complementary, I'm just going to put nucleotides on 3' end. Okay? So, now that I've got this here, okay, if I look at this, I would have a 5', so on my new growing strand I would have 5', that would mean this side is 3' on my new growing strand. Meaning, as I go this way, as this helicase unzips, it's going to expose the 3' end. So DNA polymerase 3 can come along, and on this strand, it can just start adding nucleotides one by one by one. As this unzips more and more, it can just keep adding nucleotides and adding nucleotides and adding nucleotides. So nucleotide, 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 nucleotide. Okay? So it's just going to keep adding them and adding them and adding them and adding them, and it's going to go in that direction. Okay? On this side, what's got to happen is helicase is going to unzip, it's going to lay a primer, and polymerase 3 is going to come along and it's going to add nucleotides, add nucleotides, add nucleotides, add nucleotides, because it can only go in this direction. Okay? And so it's going to do it again right here. And it's going to do it again right here. Okay? So this one has to go in this direction, where this one is going in the same direction as helicase. And we've got two different terms for those things. All right? They're called different strands. Okay? I've got a leading strand. Okay? The leading strand is one big piece, and it goes in the same direction as helicase. Okay? And then I've got the other strand that's made of small fragments. The other strand is called a lagging strand. Okay? And it's made of these fragments. Do you remember what they're called? Okazaki fragments. Okay? So it's several Okazaki fragments. Okay? So it's got several of these Okazaki fragments. And now that I've done all of this, I need to go back and get rid of those primers, because you remember what kind of nucleotides were those primers?
RNA.
They were RNA, and remember, I'm trying to make DNA, so I don't want that RNA there. All right, something's got to come along, and it's going to replace those RNA primers, it's going to be DNA polymerase 1. DNA polymerase 1 is going to come along and it's going to replace RNA primer with DNA. Okay, it's going to do something else. It's also going to proofread. Okay? Who's that? Oh, I thought someone like came in and left. All right, and then finally, so our DNA polymerase 1 came along and it got rid of all these, replaced them with DNA, but now I need to bind all these chunks together. All right, so I've got one less enzyme that does that. Do you remember what that's called?
Ligase.
Ligase. It's going to bind the fragments, okay? And this completes DNA replication, all right? And this DNA replication, it's got half of it's made of parent, half of it's made of new daughter. And do you remember what we call that? Semi-conservative. Okay, DNA replication equals semi-conservative. Meaning it's half new, half old. And, good game, America. And that's chapter 13. Okay. So if you need this, get it before I erase it. Okay. Anyone else still need this? Nope. I'll finalize this video and I'll try and put it on fourth period or during lunch. Good. Hey, let this dry for a second. I don't remember what. Okay. Welcome back to room 2. Okay, transcription. Okay, transcription is going from DNA to mRNA. All right, and it's going to happen in the nucleus. Okay, it's going to happen in the nucleus, and so now I'm going from DNA to mRNA. So it's going to happen in a similar, like, process, kind of similar to DNA replication. So just like DNA replication, what was the first step I've got to do? I've got to unzip it. I use a different enzyme to unzip it though, okay? So in this one, I'm going to use something called RNA polymerase. Okay? And this is going to unzip. It's going to unzip DNA. 10 to 20 base pairs. Okay, it's going to unzip DNA 10 to 20 base pairs at a time. Okay, and then what I've got to do is I've got to look for a very specific sequence. Okay, that sequence I'm looking for is called a promoter sequence. that's going to basically tell it start here. Okay? And in eukaryotes, I have a very common promoter sequence. Do you remember what that is? Ta-ta box. Ta-ta. Okay, that's a common promoter in eukaryotes. Okay, and so I've got this promoter sequence, blah, 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 and now I've got to have a couple things bind to it, okay? The very first thing is going to be this collection of proteins called transcription factors, okay? Okay, these are called transcription factors, and it's going to be proteins that bind to the promoter. Okay, so transcription factors, and then I've also got to have my RNA polymerase is going to come bind there also. Okay, so collectively, so this RNA polymerase unzips it. I find this promoter sequence, transcription factors bind to it, RNA polymerase also binds to it, and all of this collectively, all of this is known as a transcription initiation complex. Okay? All of that collectively is known as the transcription initiation complex. And now it's going to go along and it's going to read through the DNA and it's going to start making our mRNA strand. Okay? So all of this is known as initiation. All right? This is how it all gets started. And so after initiation, what do I do? Elongation. Okay? So during elongation, okay, RNA polymerase, I'm just going to RNA poly, adds complementary. What kind of nucleotides is it adding to this chain? Is it adding DNA or RNA? It's adding RNA because remember I'm making mRNA, so I don't want to add DNA. So it's going to add complementary RNA nucleotides. Alright, so I will not see T. Instead, I'm going to see U. Okay? T's all bad. Now I've got U in there. So I'm adding this growing chain, and this chain is going to peel off. It's not going to stay attached to the DNA. Alright, so I'm making this, remember I'm reading it like 10 to 20 at a time, and it's making this growing chain. And so that's elongation. It's going to go, and it's going to go, and it's going to go. Eventually I get to my next step. What's the third step? Termination. Termination. Okay? Okay, so eventually it's going to read a stop sequence. Okay, in prokaryotes, okay, everything stops. So in prokaryotes, everything stops. All right, and the transcript or my mRNA is complete. Okay? Eukaryotes, it's a little bit different. Okay? What?
This is how I drink it.
Sorry, I know. One of my legs is shorter than the other one. So, yeah, I know. That's life. I see life sideways. Eukaryotes. Alright. In eukaryotes, this, I'm just going to put TIC, the transcription initiation complex, is going to keep going a little bit. Okay? So my transcription initiation complex is going to continue upstream a little bit. Am I completely done with my mRNA?
No.
Am I done with everything and it's ready to go to the ribosomes? No. Now I've made pre-mRNA. Okay, now I've got my pre-mRNA, all right, and now I've got to edit it. Eukaryotes have pre-mRNA, and they do mRNA processing. Prokaryotes do not. Okay? So my mRNA processing that I've got to do. Sorry, Zaire. Okay? Homecoming's dead. Looks like my insides. Okay. There's another slip right there if you need them. All right, so mRNA processing. Okay, I've got to put some caps. So on my 5' cap, and I've got a 3' cap. Okay. My 3' cap, do you remember what I have? I have a poly-A tail, I have adenines. And remember I said, how I remember it is I know one of them's poly-A tail, one of them's a modified guanine. A comes before G, 3 comes before 5. So my 3' cap is my poly-A tail. Okay, and my 5' cap is a modified guanine. Okay, and eventually now, I've got to cut out some parts. Okay, so it'd be like, let's say I have this, all right, and in here, I go, here's my sequence. Okay, let's say this is my 3', this is my 5' cap. Okay, and I've got 1, 3, 4, and 6. And then right here I've got 2 and 5. Okay? And I'm going to say that green is going to be intron, and red, exon. Okay? A spliceosome is going to come along, all right? And a spliceosome is going to be an enzyme that actually acts like RNA. So it's a ribozyme. It's RNA that functions as an enzyme. Okay? It's going to come along and it's going to cut out a specific part. Do you remember what it's going to cut out? It's going to cut out the introns and it's going to stitch together the exons. Meaning my final product, I'll have my 3' cap. I've got my sequence right here. Okay? And I've got my 5' cap. All right? What would my number sequence be now that I got rid of my introns? I'd have 1, I'd have 3, I'd have 4, and I'd have 6. My introns are gone. So gene 2 and gene 5 are gone because they don't code for anything. That's the thing with eukaryotes. We have segments of DNA that don't code for anything. All right. So I may have this huge strand right here, but not all of it codes for it. So that's why I've got to do mRNA editing, because I've got to cut out those introns. Spliceosomes are the things that do that. They leave those in the nucleus. Exons get stitched together, and they leave. That's the end of transcription. Okay, we only have like 2 minutes left. If you have any questions, I will be here, as always, 6:30 in the morning on Monday, if you need anything. I filmed all of this, so I'll put it on YouTube. Eventually, I'll try to do that during fourth period or during lunch, so you guys have that. And you can always email me over the weekend. Okay? 35 multiple guess questions. You'll be good. It's always 35. Because I double it and make it at 100 or 70 points. Yeah. Okay? Good game, America. Good game.